I'd like to get started, and it's my sincere pleasure today to welcome our speaker, Tad Fukami. Tad did his master's at the University of Tokyo and his PhD at University of Tennessee, Knoxville, where he worked with Dan Simberloff. And he then went to the University of Hawaii, where he was an assistant professor for a couple of years before moving to Stanford in 2008. And that was terrible timing for me because I was just then finishing up my PhD and about to leave, and Tad was exactly the kind of person that I would have loved to have spent a lot of time interacting with. But I'm very happy to be able to bring him here and interact with him that way. So I invited Tad because to me he's on a really short list of people doing some of the most exciting work in community ecology, and particularly trying to understand the processes that govern community assembly. And I think a particular innovation that Tad's brought to the field is an understanding and shedding light on how historical contingency, that is, the order in which species arrive and the order and timing in which species arrive in communities can influence the ultimate composition of that community. So I think that's what he's talking about today, and without further ado, I'll pass it over to him along with this heirloom mug, which once belonged to Robert MacArthur.
Does the sound okay? Okay, well, I want to thank Rob for this invitation, and thank everyone for inviting me here and coming to my talk. Okay, so I'd like to start with thinking about biodiversity. So in an effort to understand biodiversity in ecosystem or ecological communities, there are many concepts and hypotheses that are being developed. And so daunting to think about different ways of thinking about biodiversity. But if you ask many ecologists what would be the most important or most influential concept, I think many people would say that it's still the theory of island biogeography proposed by Martha Wilson almost half a century ago now, which, as you know, says that if you want to understand biodiversity, particularly species diversity in a given community on an island, there are only two things you have to worry about: how the rate of extinction within the community, how quickly species are going extinct, and the second thing is how quickly species are going to come to immigrate, so immigration rate. If you know these two things, it's the species diversity can be understood as the dynamic balance between the two. And you can think about it, because it's so simple as you know, that you can think about any type of communities as long as you have a local habitat with the species coming from regional species pool, so that you can apply it. It doesn't have to be terrestrial community, it can be aquatic communities, or the community can be really small, so that some people apply this thinking about to understand tiny microbes and animals living in moss patches, or maybe tiny aquatic microbes and insects and so on in a, for example, pitcher plant community as local communities. But even though this is nicely generally applicable and simple, that we know that there's a lot of variation in biodiversity or species diversity that hasn't been explained well with this theory alone. So one of the important factor that we have to think about is not just immigration rate, but also immigration history. To explain what I mean, let's say that we have multiple local communities in a region, and you can think about it as a set of islands or a set of ponds or lakes. And what we know for sure now is that even if you have the same physical and chemical conditions shared by local, just for the sake of argument, that these are multiple communities, multiple physical places where the chemistry and physics are the same, and even if you have that setup, and even if you have the same set of species in a regional species pool, just by having some variation, maybe random variation in terms of which species come when relative to one another, you can have a very different set of species. So you can call it immigration history A, B, and C, and so on. And so we can think about it as a historically contingent assembly where you have to understand details of immigration history to understand biodiversity on a given island or any kind of local habitat. And this happens because of what we often call priority effects, where early arriving species, maybe these are the ones that happen to be early arrivals after disturbance, some disturbance. Those early arriving species can change the conditions in maybe in a subtle way so that to the late arriving species, that their performance will be different. And if you just change the order, things will become different in terms of community assembly. And so you can trace this back to maybe the Henry Gleason, a botanist who thought that to understand, as a botanist, to understand plant diversity, you have to think about random processes. So that was his emphasis, which is direct contrast to another big giant here, the Frederick Clemens, who also, of course, thought about succession. And to him, it should be predictable assembly because of what we might call species sorting. If you have the species, the same set of species in the regional species pool, if you have the same conditions shared by all of these local communities, if you know those things, it should be predictable because eventually there is a deterministic set of species that are dominant so that because of competitive and predator-prey and facilitator interactions, it's always going to be a deterministic species interaction that can tell predictably what species are going to dominate. So in this case, it's a predictable assembly with convergence of community composition among local habitats. And you can see, even in Campbell, that it's a dominant concept. Of course, it's deterministic succession. We like this idea because it's nice and predictable. But more, as a community ecologist, we get data from actual communities, and we now know that more, maybe Gleason was more right than Clemens. And so it's not just, so far I've talked about biodiversity, but it's not just biodiversity. You can think about functioning of communities. And those can also be affected by immigration history via effects on species composition. So just to give you an example, there's a nice study showing that the total biomass of algal communities can be affected by immigration history in the same way that if you look at the grassland plants, total community biomass can be affected by history. And also we did a study showing that there's things like decomposition and nutrient cycling driven by those wood-decaying fungi can also be affected a lot by immigration history. So it's not just fungal species composition. On top of that, we also know now that it's not just ecological problem, but also evolutionary problem, because sometimes when you have an adaptive variation or diversification happening, even if you have the same species colonizing, and depending on which species come first or late, relative to that species that may radiate over evolutionary time, you can get a lot of diversification or not. And as a circumstantial evidence, we have some studies indicating that from, for example, Caribbean lizards or Micronesian plants. And we were able to show with the actual direct experiment that this type of idea can happen. And we did that with bacterial experimental population in bacteria under lab condition. I'm going to tell you more about these bacteria later. But in any case, this simple causal relation is what we know surely happens in at least some cases between immigration history and biodiversity. And I can just replace it with a community structure just to make it general so that it's not just diversity, richness, but also composition. So this is what I like to consider as historical contingency. And then, as I said, it's not just structure like composition, but also function. And function can be directly affected by history via structure, and then function can also be feedback to structure. And so, let's go back to this. So, if you are a community ecologist, this is a scary picture. I say scary because in most cases, if you want to understand natural communities, in most cases, there's no way to know the detail of immigration history. So if history is important, we may have to give up understanding structural function in a predictable way. So you can see that there's a great temptation that we want to think that the Clemens was right, that things are predictable. But as I said, more data is suggesting to us that the Gleason was more right. And so there's a kind of frustration here where community ecology can potentially be stuck in having good general understanding. And so that's summarized by this paper. It's not so new now, but it's an influential paper by John Lawton, eminent ecologist in Britain, saying that community ecology is a mess precisely because of this type of historical contingency. He says that in this nice thought-provoking paper. And it's 1999, but I had an opportunity to review this literature recently, and this frustration is still there, that the contingency is something disturbing, and we want to avoid that and think about different ways of understanding community. So that's the state of, I think, community ecology at the moment. But the one way to make progress, because despite this type of problem, is to ask change the question a little bit. Instead of just keep asking whether we can predict precise composition of communities, specific communities. Another question that may be also equally interesting and useful is to say when historical contingency happens and when that's more important and when, under what conditions, history is not so important. And so that's the question that I want to focus on today. Basically what you're saying is that, is there anything about particular ecological context that you can use to understand the thickness of this arrow between history and structure and function? Can we say anything about context that tells us whether history is important or not? And that question is not, I'm not, of course, the first person to think about a question. There are different studies, so I'll show you just two examples. Having said that, I'm going to start with my own example. So, for example, with the protozoan communities in the lab, control communities, I was able to show that when you have a small habitat, that's when the priority effects can be important. As opposed to large habitat, there it seems like the predictable deterministic assembly seems more likely. Another example is a very nice study by John Chase, published in 2010, with the aquatic insects and plants in an experimental pond in the field, where he found that when you have a nutrient-rich habitat, that's when you have a lot of strong priority effects happening, as opposed to nutrient-poor habitat where things seem to be more predictable. And so we have this this kind of question when history should matter. That's becoming slowly more popular, and so I think we have a nice line of research going on, relatively active, although it's still relatively minor. And what's nice about this framework is that, as I just said, you can try to think about when history is important, and in addition to that, you can think about context like nutrient availability and so on, and often we see puzzling variation in terms of effect of nutrients on community structure, and the potential reason why we can't understand, for example, nutrient availability effect or habitat size effect, maybe because we are estimating the historical effects. So by thinking about these interactions between context and history, we may have a good understanding of community ecology in general. And so in my lab, so I started my lab in 2008 at Stanford, we use a lot of different systems because different systems are good at asking different types of questions about community assembly. And so I tend to be opportunistic in thinking about systems to use, but today what I wanted to do is to give you 4 answers in relation to this general overarching question, when should history matter? And I'm going to give you 4 answers based on some recent empirical and computer simulation studies to give you some flavor of what the types of general answers that you could give, potentially general answers you could give to this question. And so starting with the first one, I'm going to say that focusing on one aspect of ecological context that hasn't been so considered, I'm going to say that it's something about stability of the environment that gives you one answer about when should history matter. So that's one thing, and I'm going to use some results on nectar microbes to give you one piece of empirical evidence for that. And then the second thing is, instead of thinking just about ecological context, I also want to think about evolutionary context. And one answer I wanted to give you, also using nectar microbes as an empirical example, I'm going to say that when immigrants are closely related to one another, that's when you should think about history as potentially very important. And as a third answer, what I wanted to do now is to combine ecological context and evolutionary context, and in thinking about these two together to say that when immigrants are fully adapted to the ecological context that they are going to experience once they colonize new habitat, that's when history can be more important than otherwise. And then lastly, I want to think about feedback, I'll tell you more about what I mean by feedback here, but when I get to this answer. But I want to think about how community structure and immigration history influence each other rather than history always affecting structure. And I'm going to use computer simulation work to say that once you think about this feedback, one potential answer could be that when rapid evolution protects species from regional extinction, that's when the history becomes interesting and important. And so I'll just go through this one by one, and let's start with the local habitats being stable. So going back to this, often we think about ecological context as given. So is it nutrient poor or nutrient rich, or habitat large or small? And that's usually what, in terms of priority effect, that's usually what most people think. But of course we know, so in this case, I'm just using temperature. So is it temperature tend to be, is it cold place or warm place? But of course we know that the environment is variable. And so when you have a variable environment, is that going to tell us anything about a strength or priority effect? That's the general question that I wanted to ask because that hasn't been done so much. So basically, it's a simple question. We're just going to compare whether a stable habitat and variable habitat, and can we say anything about the strengths or priority effects that's important for historical contingency, and do we see any difference between the two? And as an experimentalist, I always want to do experiments, and to do it, we decided to use nectar inhabiting microorganisms, and I'm just putting one of the species that we see. So these are the microbes that you find. I didn't know anything about these microbes until I moved to Stanford, but somebody at Stanford suggested to me that these microbes would be a good model system to think about community assembly. And I'm just showing one of the many species that you could find in floral nectar. This is all, you can find it pretty much anywhere, as long as you have floral nectar. But using these microorganisms, we wanted to test this idea about environmental variability. So with the student that I had, Melinda, a PhD student, and Kabir, who was a postdoc with me, we wanted to use these to test this idea. So what we do is we have a local field site, and we focus on this species of members. This is a hummingbird pollinated plant that are very common in California, pollinated mainly by hummingbirds. And the reason why this is a good system, just to give you a bit of details, is that like any microbes, we know that they have a short generation time. So when you think about multi-generational long-term community assembly experiments, bacteria and fungi, those are yeast, those are convenient system. But unlike most other microbial systems that are always really diverse, this is an unusual system because it's attractable, and there's not, relatively speaking, although there are still many species, relatively speaking, it's a simple system because only a handful of species can withstand the conditions in nectar, which is mainly osmotic, really high osmotic pressure because of a lot of sugar. Only a handful of species can survive. And so we have a simple, relatively tractable system, which is also appealing for experiment. And then when you think about community assembly, these flowers are well-defined patches because, so when flowers come up, then we know that the nectar is usually sterile. And so it's a primary succession, and the only way for microbes to get into these is via actually pollinators. When pollinators come, in our case hummingbirds, they can bring these microbial species from older flower to new flower. So we can think about community assembly in a nice conceptual framework. As I said, so hummingbirds are the ones who bring species here. As you can see in this video, so if the hummingbirds go to the old flower, and if that old flower happens to have some species of microbes, when the hummingbirds then goes to a new flower, that can result in immigration, microbial immigration. And what we did is, it's fun, we go and then catch hummingbirds by mistnet, and we make them drink sugar water, and then plate those sugar water on plates, and see what species we get. And it turns out that the individuals of hummingbirds can be highly variable in terms of what species they bring. So if that's the case, you can expect immigration history to be very different among flowers. So once again, it's a nice system because you can think about historical variation in immigration history to ask questions about priority effects. And so this is our site. We have Stanford has a really nice field site here. And when you go to our field site, these are some of the numerous plants we have. And another thing we found, in addition to variation in what hummingbirds carry, is that if you look at flowers on different plants, we see a lot of spatial variation, once again suggesting that the historical contingency could be very important. I say it because we also try to measure many different kinds of environmental variables that may determine microbial composition. But we had a very hard time explaining community composition with environmental variables. That also got us thinking that maybe it's a good system to think about historical contingency. And I wanted to also mention that the reason why we were able to monitor all of these plants, about 100 plants, is because I decided to just link our research with teaching. So I teach an undergraduate biology lab at Stanford, and what they do is they take data so that and we use those data, but it's also mutually beneficial because we think we have students do an independent project, but within this general framework about nectar microbes. But so by doing that, we have a mutual benefit, I think, and I think students agree, that they provide data and they provide fresh insight into the system, but at the same time, because we work on this system as our own research project, we can provide a lot of good insight to them, to students. And so because of this mutual benefit, we were able to publish a lot of papers. We're happy about it. And on top of that, I was able to work with people at School of Education to write papers about how inquiry-based undergraduate education could be improved. So so these co-authors, I was happy to work with them. These are science education specialists. And if you're interested in not to brag, but in case you're interested in this type of inquiry-based teaching and how you can integrate, you can look at this 2-page essay about what I wrote about the summary of this class. But in any case, getting back to this question about environmental variability, what I wanted to say is that we did this experiment, and it was done by a student at the University of Toronto who spent one summer with us. And what she did is we had 4 species of microbes, 2 species of fungi and 2 species of bacteria. These are some of the most common species we get. And even though we wanted to do a real flower experiment, just to have a good control, we decided to use these PCR tubes with the nectar in it, about the same amount of nectar you get in these real flowers, and did a simple experiment where, as one experimental treatment, we had 2 yeast species introduced on day 0, and then waited for 2 days, and then introduced the bacterial species, and see which ones were more dominant. And luckily, we can just use plating to see to identify species in this case. And as a second treatment, because we were interested in effective history, everything was the same except just the order of introduction, so that we can compare them. And we also had, as a kind of a reference, we also put all species at the same time. And then on day 4, we got a little bit of nectar so that we can simulate the dispersal, so that we have a new set of nectar, fresh nectar, and then put them back, and then looked at how abundant they are. And we repeated that over about a month, 32 days. And this was done under 2 different conditions, constant temperature or variable temperature, so that we can ask this question that we wanted to ask. So I'll show you finally the data we had. So here, I'm going to plot how things were changing over time in terms of microbial abundance for each of these different treatments. And if you look at stable temperature, and if you put all species together, there are 2 species that were dominant, and 1 yeast and 1 bacteria. And the other species quickly went extinct. If you have this treatment, where you had the yeast introduced first, one of the yeast species completely dominated this community, and all other species went extinct. And so it's a simple, very strong mirror image that in this case, when you have bacteria first, one of the bacterial species were dominant, and other species, including the blue species here, went extinct. So it's a very, very strong priority effect that we are seeing here. And now you can compare what happens if you have a variable temperature. It's similar when you have all species introduced together, although the relative abundance changed. But the main difference between stable and variable temperature is that in this case, two species were able to coexist and persist, which is different. And here, the yellow may be difficult to see, but you have red and the other species of yeast, yellow species are coexisting. So if you compare these two, what you can say is that there's a very, very strong priority effect under stable temperature, and relatively speaking, it's still a strong priority effect, but it's a weaker priority effect because now in this case, the more than one species could coexist. And so obvious question is how this was happening. So we have some ideas about, we think a good idea about what happened, why we got these priority effects, and why it was stronger under stable temperature. So let me explain by thinking about resource concentration and pH. Those seem to be the key factor. And so fresh nectar sits here, relatively neutral pH, and then high resource, particularly amino acids in nectar. What happens is that for the mission cobia, this species, this is the region where they can grow. That they are good at surviving even if you have a low concentration of resources, but they are not good at low pH. On the other hand, if you have a bacteria, this is the region that they seem to be able to grow based on additional data we have. And then on top of that, what happened is that also from additional data, it seems like that if you have yeast, they draw down resources a lot, but doesn't change to pH so much. So by the time they become dominant, they get to the region where the bacteria cannot grow. And the same thing happens that with the bacteria, they reduce pH dramatically, even to about close to 2, but they don't change resources, so amino acid concentration. So this is the reason why we have priority effects, and when you have two species together, they can coexist. But this is what happens under stable temperature, and when you have a variable temperature, it seems like all of them generally have a harder time to grow. So it's a simple reason. Because of this, we have a difference between stable and variable habitat. And so it's just one simple experiment that we have, and so I shouldn't be overgeneralizing, but it's a potentially general thing that maybe if you have a stable habitat, that's the case where you have a lot of historical contingency happening, but if you have a variable habitat, it may be more likely to be, the community assembly may be more likely to be deterministic. And so this is the summary of what I just told you. But I also wanted to mention before I get to the second answer, that we've been looking also at what happens to plants and hummingbirds. Do we only have things happening at the microbial scale? And to do that, I have a good postdoc, Rachel, and Ashley was a good undergraduate student who published a paper in class 1, and Marie was a technician. With them, we were finding that if you have yeast, then even though they grow well in nectar, it was surprising to me that there is no detectable effect on nectar preference by hummingbirds or the seed set and pollination in terms of flower plant fitness. So there's no detectable effect, and on the other hand, there's a negative effect. Bacteria seem to have a negative effect in terms of nectar consumption and seed set. And so when you think about it, you have to think about community assembly because species are doing different things and they interact. And so there's a community structure being affected by priority effect, but it's also a community function that's affected by potentially priority effect. And so that's what I wanted to talk about for the first answer. And then now I wanted to talk about using the same empirical system, nectar microbes. I want to think a little bit about evolutionary context. So going back to this general picture, we thought about ecological context, but another question is where these species we have in the regional species came from the first place. So that's the evolutionary context. Luckily, as you know, there's a lot of phylogenetic information that's being built now in any organisms now, in any type of taxonomic groups now. So can we use the phylogenetic information to think anything about importance of immigration history? So that's what we wanted to ask. And so just to summarize the question, here, basically we're asking whether phylogenetic relatedness between potential colonists here, can we use that to predict strengths of priority effect? One type of naive prediction is that when you have a closely related species, they tend to be ecologically similar so that they become sensitive to historical differences because they're so similar. When you have a species A, when they come, they dominate, and if species B is so similar, they may not be able to join the community. But if you have a distantly related species, they may still interact strongly in terms of competitive and other facilitative interactions and so on. But because they are more likely to be ecologically different, the interaction can be more deterministic. So that's the kind of naive prediction that we wanted to ask. And there's no other study at this time about when we were doing this experiment that answers this question. So that's what we wanted to think about. So this is the experiment, once again, with Javier and Melinda. And there was an experimental test with nectar microbes. And in this case, we focused on just yeast community. We had 6 species that are also relatively common. These are species that we isolated from our field site. And what we wanted to do, and this is actually the actual phylogenetic relatedness of these actual species. And we wanted to use this phylogenetic relatedness to think about priority effects. So it's the same type of experiment. It's simple. We have, as a one treatment, we have one species introduced and a second species to look at priority effects. We looked to see how abundant they are, and as a second treatment, we just flipped the order. Everything else was the same. And we also have monoculture as references. And this was repeated for all of the pairwise, all possible pairwise interactions. And so I'll just show you what happened here. So I'm going to show it, focus now on just green, and just use color for a species. But so let's say what happened to green species. Here's what happened. So if you have, so this is initial density that we added to this nectar environment. And as long as the green species was the first species to be introduced, whatever second species you used, it doesn't matter. They do fine, and if you look at this water as a control, there's no difference between the two. So there's no effect of second species coming. And that's very different if you have these green species, now a second species. There is a sometimes complete suppression of the green species, but it depends on which species you use as the first species. It's going to be a busy slide, but I'm going to show you all the data here and just highlight a few examples. Here I'm comparing these two species, this species there. This is a good example of very, very strong priority effect. If you put blue species first, that's going to be the dominant species. If you put this species first, that would be the dominant species. And in this case, you cannot predict which species is going to be the dominant species unless you know what history you used to construct this simple two-species community. As the other extreme, I'll highlight this pair. In this case, regardless of which order you use, it's always the blue species that are dominant. So in this case, history doesn't matter. You can predict which one is going to be dominant because they're probably the blue species more competitive simply because of that. And so you can just use all of these data to see whether our prediction was correct, and it was correct mostly, generally speaking, that if you have a closely related species, you have a strong negative priority effect, and that's not the case if you have a distantly related species. So once again, it's just a small experiment that we're doing with the tiny creatures, the microbes, but I think there's some general implication here that if you have a distantly related species in your species pool, maybe that's when you think about deterministic assembly as opposed to historical contingent assembly when you have a closely related species. So we are not phylogeneticists, we are ecologists, and so we want to have an ecological explanation. And so far we are not really satisfied just by thinking about phylogeny. So we want to have a mechanistic explanation. So that's what we are working on now. How is it actually true that it's this ecological similarity that's giving rise to a phylogenetic pattern? And to do that, I work with Rachel, a great postdoc, much better than as a ecologist than I am. And we thought about niche overlaps, so how similar they are. So that's a classic, of course, classic concept. And this does a good job of, reasonably good job of explaining the phylogenetic pattern that we see in priority, strengths of priority effect. But what we found is that it's not really enough to think just about ecological similarity. There's a concept about impact niche versus requirement niche. Impact niche being that how much impact each species is having on local habitat and how sensitive species are. So this is to the changes in local habitat. That's often called requirement niche. If you have these three species considered at the same time, it seems like we have a better understanding of priority effect. So this is the kind of mechanistic explanation that we want to improve a little bit more. But that's the second answer I wanted to tell you quickly. And now, changing the gear a little bit, I'm going to use a slightly different, it's still a microbial system, but I have a second set of model systems that I want to talk about. And in this case, I'm going to combine ecological and evolutionary context to think about historical contingency. So getting back to this, the ecological context and evolutionary context, those are of course not separate because, for example, so I thought about ecological context and evolutionary context. But these things are not separate because, for example, the evolutionary background of species can be something related to how good they are in thriving in this ecological context that they are going to colonize. For example, how quote-unquote pre-adapted these species are to the new ecological context. Can we think about that to think about priority effects? And so that's the question that we wanted to ask, and as far as we could tell, there's no study to try to combine these ecological and evolutionary contexts. And so the question this time is, can we use poor adaptation or good adaptation in terms of pre-adaptation? Can we use that kind of information to predict the strength of priority effect? So that's the question. And we did this with another species of bacteria, Pseudomonas fluorescens. This has been a good model, a very popular model system to think about, particularly about evolutionary dynamics. And what happened is that in 1998, there is a classic paper showing that if you put this strain of bacteria into the under lab condition, so if you try to grow this in a rich medium, within 4 or 5 days, really quick, they radiate through de novo mutation into different ecotypes. And so it's a very nice system because you can ask questions about adaptive radiation in a short time in a replicated manner. And particularly if you're thinking about not just ecological but evolutionary community assembly. So we wanted to make use of this system to ask questions about combined effect of ecological evolutionary context. And what we did is I had another good PhD student, Matt Knoll, who already graduated now, with the Samantha Ford at UC Santa Cruz. We did an experiment, similar experiment, similar to the previous experiment I just told you about. And here we have ancestral genotype. These are the genotype that was just isolated from nature, and these are plant root associated microbes. So to them, this rich media that we prepare for them, that's very foreign to them. It's a foreign environment. But we also have what we call evolved genotype. These are the genotype that we cultured under lab condition for some time so that they are more adapted to this lab condition. I'll tell you a little bit more about that in a minute. But as I said, the design is really similar and simple. We have this as one treatment, and this is another treatment so that we can look at the effect of order, and we have these references of monoculture. And getting back to the evolved genotype, what we did is we have different genotypes so that they are either poorly or well adapted. We did that simply by allowing different genotypes, the same ancestral genotype, but we allow them to grow under lab conditions for just about 10 generations or 100 generations. So, and they should correspond to the degree of adaptiveness to the lab conditions. So we wanted to use these two different evolved genotypes to ask questions about whether pre-adaptation tells us anything about strengths or priority effects. So I'll give you some data here. And so in this case, I'm going to show you what happens to the number of morphotypes that arose via mutation. So this is adaptive radiation. And from these two ancestral genotypes, or ancestral evolved genotype, how many other genotypes or morphotypes are evolving under if you use poorly adapted or well adapted genotypes. So I'll show you what happens to poorly adapted genotypes here now. This is what we expected, not too surprising, that you have to be the first one to be good at radiating. Otherwise, you just get suppressed because of competition. So here, you can just focus on evolved genotype and compare the two, and you can clearly see that it matters when they come relative to the other. Each be important. But what we found is that when you have a well-adapted genotype, things are different. And so this is what happened if the evolved genotype is the second and now it's the first. And if you compare these two, just focus on the evolved genotype, history is not important. So there is something wrong, something different about poly and well-adapted genotypes. And obviously we wanted to understand what happened. And this is just to make it clear that we are thinking about the difference here, so that if you look at this case, there is a strong negative priority effect and no difference here. Yeah. So I have to be honest and say that.
Which conditions were they grown in? They were in the rich medium, or
this is under rich medium.
Always the rich.
Always the rich. So we didn't manipulate the local habitat conditions. It's always the rich media that that this well-adapted one was supposedly well-adapted. So what I was saying is that we haven't narrowed down the exact mechanism to be completely sure what happened, but based on the additional data we have and also what other people have done with this Pseudomonas fluorescens, we think we have a good guess about what happened and how to explain the difference between the two scenarios. And so I'm going to just use infamous fitness landscape metaphor. And so you have an ancestral genotype, and if you think about this landscape as a lab condition, as I said, because this is plant root associated microbial, this lab condition is not good to them. So I think it's good to assume that they are not high in terms of fitness, but as you let them evolve, they started climbing some adapted peak. But when you have a poorly adapted genotype, what happens is that they still have the potential to climb different places. But as you let them evolve and adapt more and more, eventually they find adapted peak. And once that happens, then we know from population genetics that it's difficult for them to give rise to mutants. Well, they can provide mutants, but they are not competitively superior compared to this the blue one. So they have a hard time growing in number, and we also know that the crossing of adapted poly is difficult. So once they are well adapted, the potential to diversify may be limited. So using that, I think what we can say is that if you have a poly adapted genotype, they have the ability to diversify, but because they are not so competitive, they have to be the first one to do that. Once they come too late relative to some other populations, they don't do well. So that's why we have a historical effect. But when you have a well-adapted genotype, it may not matter where they come. They're always competitive, but they lose the ability to diversify, so they don't diversify as much. And that's maybe why history is not important. And so it's just a quick overview of what we found. But I think we also have some potentially general implication here, that if you have a species that are well adapted in your regional species pool, that may be when assembly is more predictable as opposed to historical contingency when you have a set of poorly adapted species in your regional species pool. And I've been thinking about microbes a lot, so I wanted to mention a little bit about microorganisms so that you won't think that it's just microbes that we are thinking about. So I have a good collaborator in New Zealand, and there's a really good extensive data on phylogenetic information about different plants that colonize in New Zealand, the monophyletic group, and there's a lot of good data at both at the national scale and landscape scale in terms of abundance today of different species, and we were able to show using combining these two set of data that the things that we find, the general concept that we developed with the bacterial experiments could, broadly speaking, be applicable to plant communities also at the large scale, at the deep evolution at the time. And I had a student, Devin, who published a paper on that. So if you're interested in that, you can look at this paper that's published in Journal of Biogeography, another one coming up in New Phytologist. And so that was the last one. The third answer I wanted to talk about. And I have so more minutes, so I can talk about the last one. And so this one is, I think, is a most recent result that I'm excited about. I want to talk to you a little bit about computer simulation work that's related to what I was saying as feedback between community structure, immigration, history. So going back to this picture, to most of you, I'm sure this is an unsatisfying cartoon, one critical thing that's lacking, of course, is that we haven't thought about dispersal or immigration among local communities as opposed to from regional species pool. So we've been ignoring that. We should think about it differently. Another thing is that often in nature, we don't really have a stable regional species pool. Often we don't have that kind of stable pool where we can always expect species to come from outside. In this case, you have to think about the set of local communities as themselves regional species pool. So what happens to this scenario is not very well understood. I think partly because McCarthy and Wilson did such a nice job of selling this theory of island majority that we often think about mainland island scenario, but we also have a meta-population, meta-community literature, and that's not well exploited in community assembly literature, in my opinion. So as long as you think about this scenario, this alternative scenario, which may be sometimes more realistic in nature, you realize that the priority effect is a double-edged sword. What I mean by this is that double-edged sword in relation to what they do to species diversity in the region. So one thing is that priority effect could be good because when you have a variation in immigration history, then one local community has this species dominant, and if you look at the other species, other set of species dominant. So as a whole, at the regional scale, variation in species composition should contribute to species diversity. So that's good. But if you don't have a stable species pool, external species pool, eventually, if one species happens to be slightly more common in the region, then that species is going to snowball, has a snowball effect, because then that species is going to be slightly more likely to be the first earlier arriving species to the recently disturbed local habitat, and then so on and so on. So you could expect regional monopolization eventually happening. In this case, there's no variation in immigration history anymore, and that would be no priority effect and reduction in species diversity. So that's what I was thinking about as feedback between structure and history. So there's a potentially positive effect of history on species diversity in region, but there can be also feedback so that eventually a historical effect ceases to exist. Here, the question I want to think about now quickly is whether evolution plays any role. So as many of you know, I'm sure this literature now, the very exploding literature about eco-evolutionary dynamics, rapid evolution that's happening at the ecological timescale. If we think about those rapid evolution, can this scenario be changed? All of this assumes that the species are all fixed. So if we let species evolve in a rapid, relatively quickly, can this be changed so that this part, the bad part of the double-edged sword, can that disappear? So that's the general question I wanted to think about. And to do it, I'm going to just compare fixed species and evolved species. We know that if you have a fixed species, the regional maintenance of local priority effect is not possible for the reason that I explained to you. Now the unclear thing is whether if you have an evolving species, can local priority effect can be maintained at the regional scale.
No, keep going. Don't mind me, I'm going to be writing something on the board. So this is we couldn't do it empirically, so we needed to just have as a first step, we needed to do some simulation work to see whether we have any interesting idea. And so as a simplest case, on computer, we have two species, and we have a lot of local habitats in this region, and they are occasionally disturbed so that the new round of local community assembly can happen, and we didn't have any external species pool. And we are going to compare three different scenarios: fixed species, evolving species, and evolving species with trade-off. And we have two traits given to these species, how competitive they are and how tolerant they are to the toxin produced by the other species. We focus on these traits because this can be a classic case of local priority effects. No, don't worry. I forgot to announce this at the beginning. There's a party for Cat attention shots. Time at 6. Sorry.
Well, we can finish the policy on the
I don't know the address, so that's the problem.
Just ignore it.
So we wanted to use this scenario because once one of the two species become dominant in local community, then that species producing a lot of toxin that are toxic to only the heterospecific species. That's just a simple classic case of negative inhibitory priority effect. So we wanted to use that as a reference, and then now instead of thinking about, so in this case, both of the species are fixed trait. But now we wanted to just let species evolve so that they can go anywhere over time. So that's one scenario. Another scenario we used was we thought that it may be reasonable to think that there's a trade-off between how competitive they are or how tolerant they are, so that species can be individual, can be very good at competition, but they are not good at tolerating toxin produced by the other species, and vice versa. And so we wanted to use this scenario just to see what happens. So I have maybe 4 more graphs to show you, and I'll be done. So here's the regional frequency of species 1 and what's happening over time, and we always start this simulation so that both species are equal in abundance. And I'm going to show you what happened to fixed species. So each of these lines represent replicated simulation. So if you look, for example, this simulation, then they are for a while they can coexist, but once one of the species become slightly more abundant, then as consistent with what we expected, that species becomes dominant, and that's the end of story. So it doesn't matter which species, but it's always the monodominance of that regional dominance so there's no local priority. Now you can look at evolving species, the same, actually worse. When you have a common species, then maybe they adapt to more, and so the regional dominance of that species actually happens more quickly. Now things will become different when you have a trade-off. And so what happens here is that, so in all cases, two species are existing regionally. So what happened is that as one of the two species become more common, then to them, the selective pressure to be tolerant to toxin produced by the other species become weaker and weaker, and so they sort of become lazy, so that once that species become really common, then the other species, it's now easier to invade the communities dominated by species, because they can just produce a bit of toxin, and they can, the regionally rare species can take over the local community. And this is what happens if we assume that there is a common pool for dispersal and we disperse the individual. I'm going to show you just one more graph where we assume that the dispersal happens from one local community to another. In this case, things become more stochastic, and actually the regional coexistence of species becomes even better. That there's no regional alternative states now. That in all cases, two species seem to be more or less equally abundant over time on average. And so, getting back to this question, I think, yes, the rapid evolution can help if there is a trade-off in trait evolution. And so, this is what I think could potentially happen. If you have a fixed species, that the evolution doesn't happen so quickly, maybe predictable assembly happen because of dominance, eventual regional dominance, but if you have an emergent species with trade-off, historical contingency may continue to exist, so that the species diversity can be higher. I'm going to maybe skip over a little bit of this, but we want to test this empirically. We know from the Nectar yeast that there's a lot of diversity within a population, so we think potentially we can do some experiments to test this idea. One thing that I can get to talk about is transient, and I'm getting really interested in thinking about, instead of alternative stable states, which is, as you know, a very popular concept now, I think it's much better, much more important to think about what I call alternative transient states. So can we think about transient dynamics to understand community assembly? And I want to test that with theory, and there are people, collaborator or PhD students that I'm working with on that. And I also want to think about applying this. I focus on fundamental questions, but of course there are a lot of applied questions we can think about in terms of biology, invasion and restorations and so on. And so this is the take-home message that I wanted to say. We can predict when history matters by thinking about context of community assembly. So getting back just 20 more seconds, thinking about this question, the answer is can be both. That we have to think about history because it is important, but it doesn't mean that we cannot have prediction, because we can have predictions, different kind of question. A prediction about when history matters, which can also just equally important, interesting in understanding community assembly. So the long-term goal to me is to build community ecology as a historical yet predictable science. Thank you very much.
So these are questions I think about a lot as well in the context of would-be immigrants who are parasites. And in that context, the historical contingency that matters is the immunological memory, the strength and specificity of immunological memory in the hosts. And I would say if I were compiling the list of answers that people working on that angle have come up with, we would overlap a fair bit with the ideas that you have. We probably have slightly different answers, like just as an example. Phylogenetic relatedness of the would-be immigrants very often matters, but not in a consistent direction. Maybe we can talk about that later. But the one thing that seemed missing from your list that I think would be at the top of mine is the age of the habitat, the age of the host. Now, I could imagine that maybe that's a subset of one of yours, but I wondered if you could say generally, what about the age of the habitat? Where does that fit in, or is that not relevant generally?
I think that's, of course, a really nice, interesting question, and we haven't thought too much about it. So the question would be, so the obvious experiment you can do is to vary the immigration order just early or late during the community assembly, and if there is a critical time point during succession, and my naive prediction would be that it's the early time when things are different early in the community assembly, then they can go to different directions. But once the communities get to some point, then it becomes difficult to, because that's what the prior historical effects is. And so my prediction would be that the earlier you are in succession, immigration history is more important. But that's one naive, I think, expectation.
So it might be part of your stability answer, in a sense.
It could be part of that, yes.
I had a similar question, but I was thinking that it may be not only the age of the habitat and the stage of succession, but also the generation time of organisms that are living within it. Because, like, if you have like a grassland community and succession proceeds over a long time period, but grasses that change every year and plants that change every year, it may be very different from like a tropical rainforest where you have, the ages of the trees are hundreds of years, and so, the turnover is going to be much more slower.
Yeah, so that's also related to the environmental variability. Of course, the environmental variability makes sense only in the context of relative timescale of the environmental change relative to germination time. And so in our case, with the nectar microbes, we thought in California, things change daily quite a bit, low and high temperature. But in that kind of daily fluctuation, of course, doesn't matter probably to doesn't matter in the same way to plants and other microorganisms. So, but the communities consist of microbes and microorganisms, plants, and like you said. And so it would be interesting to think about when you have a community consisting of species, group of species that are very different in generation time, how does the historical contingency changes? And can we say anything about the characteristics of communities in relation to the variation in generation time of the community members? And that's something that I just thought about now as I talk to you now.
Sorry if you said this and I missed it, but to what extent are the strength of the priority effects related to initial density of the species, not just the ordering?
Yeah, so that's obviously important. So if you tend to have a population that comes in large number and everything else the same, then they should have, I would expect that would lead to, on average, stronger priority effects just because it starts with a large number. But maybe that's not what you're...
Having just reviewed the literature, do you have any sense for how general the relationship between environmental stability and, or I guess, frequency of disturbance and strength of priority effects is across other systems beyond the microbial ones? I'm thinking obviously of my islands and microbial?
To my knowledge, I don't think we don't have data yet to ask that question, but there may be some creative way of looking at existing data to ask that question. For example, if you look at beta diversity of a region, that should be partly potentially due to historical differences among local communities. And can we detect any pattern in beta diversity and environmental variability and generation time and so on? And can we see any pattern that's consistent with what we think is happening based on microbial?
When we talked about the environmental variation with the temperature, what signature of variation matters? So you showed something that looked kind of like a seasonal sinusoidal, but how does it matter, and what exactly matters about it? Should it be unpredictable? Should it be what's the?
Yeah. So we haven't done an experiment to address that question. So we have this nice model curve. It's exactly what we use in experiments. So we're using just PCR machine, the thermocycler. It's nice because you can use PCR tube and put it in a thermocycler, and we just had that program. But we haven't looked at how. So we can think about how, for example, from red noise to white noise, if you manipulate that, how random the variation is, how that might affect priority effects. But that's something that we haven't looked at. I don't have a clear idea about what will happen.
How ubiquitous is your sense of among these priority effects that you study, ones that are likely to persist for long periods of time? That is, you can imagine priority effects that don't persist. So, for example, you put one species in a community, it goes to fixation, as it were. You put another one in, and for some period of time, subject to repeated immigrations, the first species is the most abundant. But in some long-term average, the priority effect ceases to exist.
Yeah, I only have intuition rather than data on that question. My sense is that, so something that I didn't get to talk about, transient dynamics. My feeling is that the communities are actually often would eventually converge given the conditions and species set, but the transient dynamics can be can stay for a long time. That there's an eventual convergence, but the trajectory can be so variable and last a long time that history becomes important even in the absence of alternative states and hysteresis and other things that are popular now. And so that's my sense that the priority effect is not so short-lived, particularly when you have many species in a species pool. And if you have a variation in how interaction strengths on different species, once you have a subset of species, so in these experiments I have just a few species, but we've done more computer simulation work to look at many more species, and what we found is that if you have a lot of species in a regional species pool and they vary in interaction strength, once you have a subset of species established in a community, it takes a long time for that community to sort out so that eventually there is going to be a defined deterministic set of species. So I would guess that the size of regional species pool is going to determine the length of transient dynamics until eventual convergence. And we know that there are many species in the world, in nature, and so I think it can be long term.
All right, thank you very much, Ted. So the address is
123 Valley Road.
123 Valley Road.